All right, thanks, Peter. I hope everyone can hear me and see my screen. Welcome, and on behalf of NCBI at NLM, I would like to welcome everyone to our webinar this afternoon. I'm Valerie Schneider, and I am the lead for the Comparative Genomics Resource, or CGR project, and I want to thank you in advance for taking the time to join us. We really have two primary objectives for this webinar. First, to tell you about the CGR project and why it matters, and also to learn about your experience with comparative genomics and how CGR can help. So to accomplish the first objective, you'll hear me describe CGR and its potential benefits to researchers. And to accomplish the second, we're going to have a few polls and some discussion at the conclusion of the presentation. And I really encourage you to be a present and active contributor during this webinar. So to start, we're going to just ask you a few questions about your experience with comparative genomics so we can get the most out of our time together today. So Peter, if you want to go ahead and start with the first poll.
Yep, Valerie, so this is the first poll asking about your experience with comparative genomics. And so I'm launching the poll, and you should be able to go ahead and vote, and we can see how many people have voted, and then we'll stop it once we get some responses. Okay, looks like the majority of people have responded. It looks like we kind of messed up the poll. It looks like we had the same choice in there twice. So we really have only moderately experienced and very experienced options available for you. I'm going to share the results so we can see. So the majority of people called themselves moderately experienced, but we didn't do that poll quite right. Okay, I'm going to go ahead and stop sharing that. And then Valerie, you want me to go ahead and put up the second poll?
Yeah, if you can go ahead and do the second poll, that'd be great.
And this is about activities that you perform in comparative genomics work. And this is a multiple choice. Okay, the majority of people have answered this, so I'll go ahead and end the poll and share the results. So lots of different activities that people are doing, a lot of downloading and annotating and assembling genomes. Okay, so I'll go ahead and stop sharing that for now.
Thanks, Peter. And thanks everyone for sharing that feedback. And we'll try to keep these results in mind as I move through the presentation today and also as we get to the Q&A. So I'd like to start with a few questions for you to think about while we're going through this presentation. As I mentioned, we'll be opening for questions and discussion at the end, and I hope these questions here will guide your thoughts, for this area. Community collaboration is critical to CGR project success, and feedback from the genomics community will inform improvements made to existing NCBI data, tools, and interfaces, as well as new developments in these areas. So these questions are really an area of dialogue from which we'd particularly like to hear from you. We're interested in how your work relates to the CGR and how it might have the most impact on your work. We're also interested in learning from you about the different ways in which you'd like to stay engaged with NCBI and the CGR project, both to stay up to date about current work, but even more importantly, so you can inform us about how CGR can best meet your comparative genomic research needs. I'm going to cover the following items associated with CGR. We'll talk about why research organisms matter, the problem CGR is trying to solve, and how we're working to meet that challenge. I hope that as you learn about CGR today, you'll consider how it relates to your work and how you might be able to inform the project. Today, you'll hear me talk a lot about research organisms. And to make sure we're all on the same page, I want to make clear that when I use the phrase research organisms, you recognize that I am talking about the broad collection of all eukaryotic taxa on which research is done. And the value of research organisms is twofold. They enable us to understand basic biological processes, and additionally, they help us understand human disease. The power of research organisms is realized through comparative analyses to systematically explore and evaluate biological relationships. And comparative analyses have been used to study areas as diverse as host pathogen interactions, including those involving the SARS-CoV-2 virus, and cancer. And it's also informed vision studies, our understanding of biochemical pathways, pharmacogenomics, and drug development. And even the field of xenotransplantation, recently exemplified by the transplant of a pig heart into a human, has also been enabled in part by comparative analyses of immune regions. So we are now poised to make bold new strides through comparative genomics as advancements in sequencing technology and algorithmic methods have democratized the generation and analysis of genomics data, resulting in an explosion in the number of organisms used for research and a rapidly expanding collection of sequenced genomes. Now, while research organisms offer great promise, the current landscape of individualized data resources for the greater universe of these organisms prevents investigators with several limitations and challenges when it comes to using the data. For example, there are multiple different user interfaces. Only a limited number of organisms have robust corresponding data resources. The data and applications may be siloed, and users must typically download data to apply tools. So there's really a limited scalability in the current approach. Considering those challenges, NCBI established a multi-year project at the National Library of Medicine to maximize the impact of eukaryotic research organisms and their genomic data resources to biomedical research, called the Comparative Genomics Resource, or CGR. NCBI, as part of NLM, is charged with leading the development and engaging genomics communities. The CGR project will facilitate reliable comparative genomics analyses for all eukaryotic organisms in collaboration with the genomics community. And as we proceed with the CGR project, we are keeping in mind the broad and diverse base of users who study or make use of comparative genomics in their work, and we value feedback from all of them. So CGR's mission, as I mentioned, is to establish an ecosystem that will facilitate reliable comparative genomics analyses for all eukaryotic organisms in collaboration with the genomics community. This image here on the right highlights this ecosystem. In the middle is NCBI with its data, tools, and interfaces that can be used for comparative genomics. In the outer ring, you have community-provided resources that can be brought into the NCBI ecosystem through standardized connections, illustrated by the arrows. Having an organism-agnostic central connection point with core content that can also offer standardized connection interfaces to community-provided content has the potential to bring together data from the expanding universe of research organisms in a way not previously seen. And through the establishment of this ecosystem, the CGR project will then maximize the impact of eukaryotic organisms and their associated genomic data components to biomedical research. So we believe that the CGR project will offer the community several benefits, which are outcomes of the approach that we're taking. We start with a foundation of core genome data available in the archives at NCBI. Because more than half of assemblable genomes submitted to GenBank today are contaminated with foreign sequences, and as much as 80% lack functional annotation, a key component of comparative analyses, as a part of the CGR project, we're developing publicly available NCBI tools for data producers to use prior to data submission that will address both these issues. Their use out in the community will further increase the quality of this base of data, as will NCBI's own use of them in our work to improve existing content. We're also curating content, adding features to make it more usable for comparative analyses. Next, with CGR, we'll offer new and improved comparative genomics tools. In addition to the tools to increase the quality of submitted genomic data, NCBI through CGR is improving existing tools and developing new ones to provide you with a seamless experience in exploring, analyzing, and retrieving eukaryotic genome-related content. These include new BLAST databases and a new comparative genome viewer, as well as new and improved web and programmatic interfaces to find and download these types of data. The CGR project will also offer the benefit of scalable analyses. Both the number of genomes in GenBank and the species they represent are growing annually and at an increasing rate. To help you take advantage of these data, NCBI is developing content and tools to support emerging big data approaches to comparative genomic analyses, such as facilitating the creation of AI-ready data sets and cloud-ready tools. I also want to emphasize that the CGR ecosystem will offer opportunities for new discovery amplification. By providing equal access to genomic data and tools for all eukaryotic research organisms, including those not represented by organism-specific resources, NCBI is increasing their potential contributions to research. This amplification also comes from CGR efforts to enhance NCBI-held content with community-supplied content and to connect NCBI resources with community-provided resources. So put together, by maximizing this impact of eukaryotic research organisms to biomedical research, we're really offering new possibilities for scientific advancement. This next slide here provides a closer look at the CGR ecosystem that I previously mentioned. On the outside of this image, you can see what we mean by community-provided resources, which include such things as organism-specific knowledge bases, genotype-phenotype relationships, variation data, and even image collections. The smaller circles here with arrows illustrate that NCBI, through the CGR project, will provide standardized infrastructure and tools for data connectivity that enable opportunities for tie-in of community resources and content to data and tools at NCBI. Listed in the inner circle are some of NCBI's data and tools that can be used for comparative genomics. On the left side of the slide here, I've highlighted several specific resources under development or improvement as part of CGR. These include data resources, such as NCBI datasets, which lets you easily gather genome-associated data from across multiple NCBI databases. It also includes analysis tools, like CGB, a new comparative genome viewer that allows you to compare two genomes based on assembly alignments. You can learn more about CGB at an upcoming NCBI webinar. And a tool we've recently released to improve data quality is a new foreign contamination screening tool that detects contaminants from foreign organisms in assembled genomes. A recording of a recent NCBI webinar describing this fast and highly sensitive tool is available now, and that's available now in GitHub, can be found on the NLM NCBI YouTube channel. So I'll now just share a couple of examples of other recent developments that are part of the CGR project and are already available to you. NCBI Gene is an example in which externally sourced content has been added to NCBI records as part of the CGR project. NCBI automated a process to import descriptive information about genes from the Alliance of Genome Resources for several organisms, including C. elegans, Saccharomyces cerevisiae, zebrafish, mouse, rat, human, and Drosophila. And as shown in this slide here, the summary section of the Gene Full Report page has links to gene pages at the Alliance, connecting this resource to NCBI. And in the cases of genes that don't have a RefSeq provided summary, NCBI provides the textual gene descriptions from the Alliance of Genome Resources and gives attribution to the source. NCBI also automated the import of publications from several model organism databases associated with the Alliance and has created corresponding gene PubMed links. And these additional publication references are included on NCBI gene record pages in the bibliography section. We've also developed new web interfaces informed by extensive user testing in NCBI datasets as part of CGR to help you find and retrieve genome data. In the search bar at the top of any NCBI page, you can type in an organism name, and you'll get a straightforward knowledge panel that connects you to relevant data and tools. Clicking on the organism's name will then take you to a new organism page. On an organism page, you can browse all genomes, find information for the reference assembly, as well as annotation and genes. And I'll also give a quick shout out here too to our newly redesigned genome assembly web pages, which include assembly statistics, sample details, as well as information on assembly methods, annotations, and revision history. And they also offer links to related resources like PubMed, Bioproject, GDV, the NCBI Genome Browser, BLAST, and other tools. Returning back to the species page, you can also investigate the availability of genome data at different taxonomic levels by clicking this Browse Taxonomy button. Additionally, the download button here provides fast access to the download of a user configurable data package with sequences, annotation features, and metadata reports for the reference assembly. Users can also create download packages for other genome assemblies and for genes. And for those of you who want programmatic access to genome and gene data, we also offer command line tools and a RESTful API. A recently developed CGR associated analysis tool is ClustredNR, which supports comparative genomics analyses. NCBI created this new BLAST database in order to provide reduced results redundancy as well as faster searches. It's derived from the default BLAST protein NR database by clustering sequences at 90% length and identity. And it really lets users see hits across a wider range of organisms and evolutionary distances. This image here on the right just shows you the choose search set option section of the protein BLAST submission form where you can find this new database. And to help you assess the new database, we also offer you the option to run your searches simultaneously against the clustered NR as well as the standard NR database. So you can access these as well as other CGR related data and tool developments that I don't have time to cover in today's webinar through the CGR website. And I'll also point out that at this website, you can sign up to get emails about updates. On that note, this slide shows you some suggestions for how you can help shape what comes next for CGR. We have really thick skin at NCBI, and we're eager to hear how we can improve our data tools and resources to better support your research needs. You can email us directly at the address shown here. You can also sign up for the CGR Updates email newsletter to stay up to date on the latest news and information. And listening sessions are opportunities for those currently working on eukaryotic comparative genomics to talk to us about how CGR might help them. And lastly, you can go to our website to learn more about CGR and the data tools and resources that make up its ecosystem. So I'm just going to pause here for some Q&A, and Peter, let's see if there's any questions.
Hi, Valerie. Well, right now there are no questions from the audience, but while I have some questions that I think are useful to ask, so I'm going to ask those. And then while I'm doing that, if anybody has some questions, please feel free to type them in the Q&A pod. So here's some questions, Valerie. CGR offers NLM great opportunities to make actual use of content and tools developed in the community. What do you think some of the low-hanging fruit in this area might be? And what challenges do you foresee as you move forward in this area?
Thanks, Peter. That's a really good question. I think we have a lot of expertise in the community in areas of targeted gene annotations in assemblies. There's also a lot of information about organism descriptors, as well as content that refers to not just the annotation, but descriptors of genes that, there's a lot of expert information in that space. There's also, I know, a lot of work going on in the area of tool development, also in the area of annotation as well. So those are all places that I think we're interested in really connecting with the community. I will say that, however, like one of the challenges that we face is that CGR and Ncbi is a resource that is working at a very large scale. And so content that we take in or tools that we want to learn more about and connect with people on, one of the things that we'll be learning is how well are these things suited for adoption at a production scale. And so there may be cases where we can't adopt them directly, but we may stay engaged with those groups to work with them to figure out how we could make something like that work at production scale.
Okay, thanks, Valerie. And here's another question. How will you balance the feedback you get through engagements with the roadmap that you already have for your planned work?
That's a great question too. So as I mentioned, I think many times in this webinar, we are really interested in user feedback. But obviously you don't tackle a project like this without having some kind of plan. But I'll emphasize that we're using a development method for this project that allows for regular inputs of user input. And so as that feedback comes in, every cycle that we go through gives us an opportunity to look at our roadmap and assess, is that feedback telling us whether we're on the right track or not? So it's not a case of we intend to produce CGR, get your feedback, and then be like, oh gosh, we need to start over. We want to have this feedback as we're continuing to build so that we can pause our development and look to see whether we need to reprioritize some things in terms of moving them up on our timelines, or if there are things that we've actually missed on our timelines.
Okay, thanks, Valerie. Also, can you talk a little bit more about the reception that CGR has received so far, especially when it comes to the other model of organism databases? Yeah,
thanks, Peter, because I know that we may have users and listeners on this webinar who work with a number of other resources for comparative genomics, many of which are organism specific. I will say that as part of the CGR engagement and the ecosystem that we're developing, we are engaging with those groups. We are talking to the Alliance of Genome Resources. We are talking to, as I mentioned already, we are talking to other groups there. And so far the reception has been very positive because we recognize that we each bring different things to this ecosystem. And a lot of our ongoing discussions right now are how to ensure that we don't have overlap and how that we can make sure we're using each other's strengths to make that ecosystem something that is beneficial for the user community writ large.
We have a couple of questions in the Q&A that are really kind of specific help desk questions for Ncbi, and I don't want to spend time answering those. We can probably answer those either offline if you write to our help desk, or I can put them in the Q&A document. But there is one that's kind of a broader one. I think it's about data sets. And it says you're implementing the data hub page, but there's some features from the legacy page. And I'm not sure which legacy page this person means, but maybe some of the old genome pages or from the assembly pages. Are those going to be implemented in data sets? Genome pages is specific, he clarified that.
Yeah, so that's a great question. We are doing extensive review of how the legacy pages have been used, and we're also doing a lot of user feedback and testing on the new pages. And again, if you want to participate in any of that, please, again, you can email us directly at cgrnlm.nih.gov to make sure we hear your feedback on that. we are making sure that those most heavily used and valued features that are there in the legacy will be available. But I will say it will be a little bit of a new experience. And so while some of the things that you might have done in one way in the old system, in the legacy system, they'll still be there in the newer system, but the approach might just be a little bit different. But we are very cognizant of what is on those old pages, and we're watching them carefully. And in the same way, as I mentioned, the whole CGR project is rolling out sort of in incremental phases with user feedback. That's also what we're doing with data sets. We want to get the most heavily used things out to folks as quickly as possible so they can take advantage of the new space, but it doesn't mean that just because a page has come out, we're done working on it.
Okay, great. So please, if you think of any other questions for Valerie, put them in the chat. And like I said, if it's a specific question about an NCBI resource, like BLAST or something like that, please write that to the help desk. What might be useful now is we can ask some questions to the audience and see if you'd like to answer those in chat. And also, we would like to know if you think the purpose of the project is clear. So it would be useful if you could put answers to that question in chat, and let's see if anybody has anything to say about that. There is another question, Valerie, but I think, yeah, I'll go ahead and ask this one. Is CGR working directly with the All of Us research program, which is really more about human, but I guess this is a useful question to answer.
So that's a good question, because comparative genomics is relevant to human health, and that's here at NCBI that is our mission. And so while we haven't yet engaged directly with them, we are very cognizant and aware again of the human health related projects and our oversight committees, a couple of which are listed on this slide here. We have representation from folks working in those areas, so that we will be reaching out and connecting with them as well.
Okay, and then somebody just asked a general question, how you can learn more about CGR, and Sarah put the URL directly to the Comparative Genomics Resource in the chat for the meeting, so that's a good place to start.
Great, thanks. All right, then I guess I'll say, I want to conclude this presentation by thanking the many people that have been involved in the work I told you about today, and these include the team leads at NCBI here, as well as their development teams, our leadership at NCBI and NLM, as well as our oversight committees. And CGR really takes a village, and these people are working tirelessly to make it a reality. So I want to thank you for your time and mention that we hope to continue hearing from you, and we're eager to keep you informed on the latest with CGR. And so some ways to do that are shown on this screen here. And we're also about to put up a final poll so that we can better understand what types of CGR engagement opportunities might be most valuable in order for you to inform its development. And just a quick reminder that the best way to keep informed on upcoming events and opportunities is to subscribe to the CGR newsletter. So Peter, if you want to go ahead with that final poll.
This is about opportunities. What types of CGR engagement opportunities might be most valuable for you? These are various ways that we can reach out to you, and you can talk to us. And if you have any options that aren't listed here, you can reply in the chat, and I know somebody did on an earlier question. Okay, I'll go ahead and end the poll and share the results. So lots of interest in all the various options that we're offering here.
Fantastic. For those of you that selected listening sessions in the poll, I want to point out that we have 2 upcoming sessions for those currently working in comparative genomics. And so we hope to hear from you in those sessions to learn your perspective on the genomics tools and resources you use, how well current NCBI resources meet your needs, and resource gaps and barriers in comparative genomics that NCBI could address. And that information, I believe, is also showing up in the chat for the registration links. So with that, I'll conclude what I have to say for this NCBI Minute, and thank you again for your time.
Okay, so that will conclude our webinar for today. So thanks everybody for coming, and I hope it was useful for everyone.